Hello, everyone, and welcome to Field Notes. My name is Casey Latta, and I am the MLRA office leader in Lincoln, Nebraska, and the Field Notes regional representative for the North Central Soil Survey region. As a regional representative, I serve on the Field Notes Review Committee. The Review Committee solicits and selects topics for each webinar. We have selected two exciting topics for today's live event, but first, let's go over a few housekeeping items. This is a Microsoft Live event, not a Teams meeting, which means you are joining today's webinar in listen-only mode. We encourage you to ask questions at any time using the Q&A panel. The Q&A panel should open by default. However, if for some reason your Q&A panel is not open, simply click on the question mark icon located in the upper right side of your screen. For closed captions, turn on the live caption button located in the lower right corner. Today's session is being recorded. Recorded sessions are available in Teams on the Field Notes channel and the National Soil Survey Center's YouTube channel. Again, thank you for joining us. I hope you enjoy today's session. With that, I'd like to turn it over to the Field Notes moderator, Dave Hoover, to tell you a little more about today's webinar. Take it away, Dave.
Thank you, Casey. Hi, folks, and welcome to Field Notes webinar number 20. That's quite an achievement. I don't know that we knew that we'd go 20, but we're still strong, and I see us going 20 more after this. It was mentioned about the YouTube channel. What a wonderful thing to sit around the hearth at Christmas time and look at your favorite reruns with your family of Field Notes. Let them see some of the things that not only that you do, that they hear about, but what people are doing around the nation. Also, what better Christmas gift to give to a couple of hundred soil scientists than volunteering your own time to do a presentation on Field Notes. It's a great opportunity to share what you do out in the field or in the lab and let others see what's going on and have questions, have some good dialogue, and it just takes a few minutes of your time, well spent. So think about both those things this holiday season. With that, we'll get into our presentations today. We have a couple of scientists from our research staff at the National Soil Survey Center presenting today. And the first of those will be Beaudette from our Sonora, California office. And we'll be talking on quantitative use of soil color, supporting the development and presentation of soil survey data. Dylan, the floor is yours.
Yeah, thanks, Dave, and everyone else for setting this up and inviting me today. I'm a research soil scientist on the Lincoln staff, and I'm going to talk a little bit about soil color, the way that we've used it through time, and how maybe we can use it on into the future, and then what I think are some exciting ways of developing a set of maps and material for scientific communication, right? So for relaying complicated scientific findings and interpretations to the general public, which I find to be a really fascinating and rewarding part of what we do here. So I'm gonna turn off my camera and switch to the slides, and feel free to ask questions. There's a lot of material here to cover smashed into a small amount of time, and so I'm more than happy to follow up one-to-one or one-on-one with anybody. So, like I said, this is an expanded version of a previously given talk from a couple months ago, but I do want to take this time to acknowledge the co-authors, so Chad Ferguson, Jason Nemechek. But this really is an ongoing project, kind of a long history, and so this work has been supported by a number of folks, colleagues, cooperators at universities, various supervisors over the years, people who contribute to our packages, color scientists, and then really, it goes without saying, right? When you put something together for, say, all of CONUS, you're talking about 100 years of soil survey efforts. The curation of our official series descriptions, and for this latest set of soil color maps, we're talking about all of that hard work that went into creating updating, correlating data map units, linking pedons, and then the final presentation of that information in a product like GNATSCO. Right, so this is really all of our work that's been distilled down into a couple of pretty pictures. A very brief history here. Like I said, this tinkering with soil color, my interest in soil color goes back a long ways, even before 2005. I think it was in a soil genesis and morphology class at UC Davis when I first became aware of the fact that soil color varied in depth and space and time, and that is really what sucked me into soil science, a graduate career at UC Davis, and then eventually soil survey. And so you can see this truncate, or rather this condensed timeline here, represents a number of events on this progression from trying to connect patterns in space and landform to soil color at Pinnacles National Monument during that soil survey, to using a soil color database developed by Tom Reinsch, and then the integration of color and sketches into soil web through the official series descriptions on down through a series of maps. So I want to quickly show you this progression of maps from 2014, and then we'll jump into the main content of the talk. So this is zoomed into an area of Baldwin County, Alabama, right? So this represents the first set of soil color maps made for CONUS. This is StatsCo. The spatial data comes from StatsCo. The colors come from the official series descriptions. So this was from 2014. This is 2018, where we used Surgo, StatsCo, and then colors from the OSDs. Chad Ferguson came up with a really clever way of filling in gaps. So the gaps, as you can imagine, are going to be miscellaneous areas and higher taxa components that you can't tie an official series description to. So even though this looks kind of pixelated at the extent of CONUS, the maps were, I think, beautiful. This time around, we're using GNATSCO, the 30-meter version of GNATSCO. Right, that's the compilation of Surgo, StatsCo, raster soil survey. And this time we're using component pedons for colors when they're available, OSDs when they're not, and then some special colors for miscellaneous areas, and then using MLRA colors. So the most frequent color for an MLRA as the final sort of backup. And when you put those data on, drape them over a DEM, the colors, the landscape positions, the landform shape, all of those things just really pop out. And some people might say, well, this is just a pretty picture, just tinkering with colors, but there's a lot going on here, right? These maps really represent a distillation of field lab database, OSD work over the generations. You know, if you've ever read about or tinkered with or taken any of the statistics for soil scientist courses, you know, this represents a projection. So imagine a very complex multivariate data set, which is soil survey, that's been flattened, simplified down into a single map that represents soil genesis and geography, which I think is a very effective way of communicating about what we do. Since we're all soil scientists here, I couldn't talk about this at the World Congress, but I wanted to briefly point out that for each GNATSCO map unit key pixel, right, so for each 30-meter pixel tied to a map unit key, the colors are all linked up to component keys. So first with a component pedon if it's available, then with the official series description if it's not matched on the component name. The most frequent color by component percentage is selected for that map unit key. And then there are some special colors for miscellaneous areas like water, dumps, pits, mines, playas, rock outcrop. And then we also use a special color to signify basically a below contact condition. So for representing a color below the depth to paralithic or lithic contact. I'll briefly mention that none of this would have been possible. These really detailed high contrast maps would not have been possible without the really incredible degree of component pedon linking that has been done over the years. I suspected that there would be a lot of component pedons linked, but I didn't suspect that there would be this many. Basically, these colors represent the fraction of an 800-meter grid cell where you have a component pedon linked to the components of a map unit. In short, yellow colors mean nearly complete coverage of map units, components, and component pedons covering that area. I think this is just really incredible. All right, so switching gears just a little bit, right? Soil color is a big part of what we do. It's the first thing you see in the pit. It's an incredible integrator of processes and properties. It may not be the final call on, say, the depths or the transitions between genetic horizons, but it's a big part of that process when we sit down to describe a pit. And when we're describing soil color, when we're talking about soil color, we use the Munsell system. It's roughly based on a perceptual model of how average humans perceive color. And it's also a convenient bridge to other color systems. So the CIE lab color system, the sRGB color system for displaying colors on the screen. There's linkages between these different color systems that we can take advantage of to describe or to communicate about the different aspects of soil color, the way colors relate to each other, and the way in which we cook down, say, 20 pet-on observations and their genetic horizons into a single representative color. Now, when I talk about contrast, I'm talking about comparisons. We can talk about mixtures of color, sort of the inferred synthesis of, say, organic matter, so pigments in the soil, organic matter, iron oxide, or iron depletions. And then we can talk about the aggregation of colors and then maybe signatures of colors. So in short, soil color is not the central focus of field description and the way that we present our data, but it's behind the scenes. It's a very important tool that we all use and understand. I've used this slide for a number of talks, so maybe you've seen it before, but I think it's an important reminder to myself and to others that when we're describing a soil in the field and when we're modeling soil color on the computer or at our desk or presenting it to someone, say, in SoilWeb, we're working with these abstractions and generalizations that are fairly removed from the original, from the original soil profile. So it's important to remember to not take this stuff too seriously, but remember that science is largely based on models and approximations. And the better and more efficient those approximations, maybe the better we become at communicating to each other and to the public about complicated systems. Okay, so there's a little bit of what looks like computer code in many of my slides. This is R code citing R packages, SoilDB, AQP, and specific functions in those packages. This is a reminder to myself and to all of you that everything in this presentation can be recreated using a little bit of R code interfacing with our databases. Okay, so this stuff is accessible with just a little bit of a learning curve. Intuitively and also explicitly, we think and talk about color comparisons, right? If you're describing, say, the differences between moist and dry colors, these are colors associated with the Clarksville soil series from Missouri. Usually we're talking about differences between colors or contrast in the context of redoxomorphic features for wetland delineation and criteria, or for describing sort of the intensity of this, say, iron cycling regime, or the intensity of the manganese stains on pet faces or rocks or something like that. And Soil Survey Technical Note number 2 talks about our soil color contrast classes. You might recognize, say, distinct, faint, prominent. Those are qualitative descriptions of the, say, change in hue, value, and chroma between a reference color and some color to be compared. This funny looking thing here can be read as the delta E00 or the CIE 2000 color contrast metric. This represents a quantitative description of the perceptual difference between two colors. And that's what these numbers represent here, say. So the delta E00 between 7.5 YR 5/6 and 6/5 is 9.83. This is a roughly linear scale of average human perception of the differences in color, right? So bigger differences as you shift between chips in value, smaller perceptual differences as you shift between chips of chroma or hue. One other sort of demonstration of this, and again, this is available in that AQP package for R. We can think about a gradation in, say, a reduced matrix or a matrix color, possibly reduced, and oxidized iron masses, right? F3M in field vernacular. As you might expect to find in a gradient from, ever so slightly reducing oxidizing conditions leading to the formation of oxidized iron masses, to a situation where you have an intense or prominent sort of representation, manifestation of these things. So I made this as a bit of a calibration for myself and for others to understand how varying, say, the Munsell chroma affects the contrast class and the delta E00. There's a lot to unpack here and talk about, and it's something I'd like to write up and discuss further on. But again, this is a demonstration of how we use color internally. We can also talk about simulated subtractive mixtures of pigments, based on reference spectra, proportions of different pigments we know are in the soil, to come up with reasonable estimates of colors that you might expect by mixing them in known proportions. Soil color aggregation refers to cooking down lots and lots of data into a single representative set of colors. So in this case, these are the top 50 soil series per soil order. These are moist colors for near the surface and below the surface. There's a lot going on here. I think many of you will find, for example, the ultisols have really high chromas, red colors. The vertisols have dark colors. A lot comes out of this representation here. Generating aggregate colors for soil order is more of a PR stunt and not necessarily something we use on a regular basis. But you could imagine doing this for, say, a component concept or a series concept. You can do the same sort of thing. Say you collect up all of the redoxymorphic feature information from our NASA's add-ons. You can come up with representatives, so something called the L1 median. This is the multivariate median of soil color. It's a little bit different than the average. This is sort of the central tendency of, say, all of the manganese concretions described in our database. So these tools allow us to synthesize and summarize, and also describe differences, say, between entire profiles, right? So with using some of this functionality within the AQP package, you can actually arrange and search for soils based on sort of the colors and the position of those colors by depth. Okay, so now the fun part, right? We have to communicate all of this information to each other and to the general public. And I think soil color tells a really interesting story. This is one of the first things I do when I am teaching in the field. I line up the material from Genetic Horizons, and if there's some sort of rock underneath, I shove that down at the bottom, and this is where discussion really starts. So we recognize that there are many different pigments in the soil, and understanding or being able to identify some of the dominant pigments speaks to the processes, maybe limitations, possible interpretations of that morphologic description. So these right here represent a kind of far away look at the CONUS level look at the 2022 soil color maps. You can't see the detail at this scale, but they're fun to look at. There's a lot to talk about here. Up close, they look a little bit more interesting, right? I've arranged sketches of the dominant, the most frequent official series descriptions, or the series within this little viewport here, and the little green circles represent those depths at which slices are displayed here. It's really interesting to see how the depressions really and the drainage classes really pop out. Okay, again, another reminder of, if you take a strip through central Minnesota, Mississippi River, and you just look at this progression from 2014, 18 on down to 22, this represents an order of magnitude increase in detail. This is a broader look at central Minnesota here, where you can see the Mississippi River, the floodplain, the glacial features, I'm not from here. I haven't done any soil surveys here, but with a couple of minutes of reading official series descriptions and map unit descriptions, there's a lot to say about this area here. And you can arrange the official series descriptions. That's what these sketches represent here. These are OSDs in sort of thematic form or graphical form. You can arrange component pedons. Little green dot is again a reminder of that map that we were just looking at, was a slice at 50 centimeters. And these are arranged roughly based on a two-dimensional hill slope position. All right, so this is, we're looking at a drainage sequence here, upland, lowland. And as you would expect, or at least as you would hope, or as soil genesis, theories of soil genesis predict, there is a progression of brighter chromas, lower chromas along this drainage sequence. Taking a look at the eastern side of the San Joaquin Valley here, this represents, so this is in California, a series of inset alluvial fans related to glacial outwash cycles. So as you move from the bottom of the fan, the distal end here toward the center of the valley, on up closer to the foothills where you have bedrock controlled soils, you're looking at older and older surfaces. And if you can bring up some of the OSDs from this area and align them, say, by their subgroup level, taxonomy, annotated with, kind of where they sit on that sequence, there's a lot to say. There's a lot that could be said and communicated to folks that don't necessarily have a background in any of these specific topics. Right? These are real color, as long as you're not colorblind, the morphology and the geography is fairly straightforward. This is a fun example from south central Iowa, where you've got this highly dissected till plain, and the orange represents a fairly interesting situation where you have the Shelby series and you have basically the bits of a paleosol peeking out on these eroded shoulder positions. Now that orange isn't visible in the OSD, unfortunately, but it is visible in the component pedons that are linked to these map units and components. And then, essentially this diagram on the right here from the OSDs and from a distillation of Surgo two-dimensional hill slope positions, the drainage sequence and the soil morphology is a fairly, I don't want to say simple, but it's a fairly coherent picture of a soil system. Another example from Texas, where you have lithologic control on soil color, essentially older surfaces on limestone, vertisols on marl and chalk, and then soils formed on more resistant forms of sandstone, giving predictable differences in soil color. Lining up the OSDs, we can kind of see the shallower soils on the more resistant bedrock, the older surfaces with the bright red, 2.5 YR hues, and then, some interesting features like reworked Aeolian sands with these lighter colors, higher value, and yellower hues. One more example, this one from eastern Pennsylvania. This is one of my favorite, basically these tortured ancient rocks and, sort of drainage systems that are reworking material. Largely, the colors here are largely controlled by the presence or absence of a fragipan, but also, different rock types here in poorly drained metasedimentary rocks versus diabase versus limestone, each with their characteristic colors. Well, that was a lot of material, and I'm happy to follow up on all of these topics. The maps are all available online in a number of different places. And, my co-authors and I would really like all of you to be able to use these maps in whatever you're working on, either, update work, initial mapping, communicating with the public, or just a nice background for Ag Handbook 296. So please let me know if you have any questions or comments. Thanks.
Thank you, Dylan. So a couple questions did come in. So take a quick deep breath and I'll read off the first one. For the soil colors of the U.S. poster or a similar color map, are those colors shown for an area determined as the most frequently occurring soil color between all of a component's horizons down to 200 centimeters, or from a more specific zone?
That's a great question. And just like these maps here, the poster, which represents the 2018 vintage maps, the poster represents a specific depth slice. And within that depth slice, it represents the most frequent color within the map unit.
Okay. The next question is, are the graphics from your slides like the color comparison for MAT versus F3M chart or RMF colors moist color chart available for download anywhere?
Yeah, well, let's talk about it. You know, this presentation and much of this material represent an evolution of ideas and diagrams and maps that you know, really need to be formalized, I think is the right word. Zamir and Chad and Jason and I are thinking about publishing in one of the, you know, probably one of the nature journals, because this is kind of a, you know, flashy pictures and stuff. And we may even try for a short article in National Geographic, because this stuff is just so visually interesting. But yeah, I'd be happy to share all of the material. Just contact me directly.
Great, thank you. We'll give just a minute for any other questions. I have one while we're waiting. So I was just saying that, you know, when LiDAR came about, we saw new things on the landscape that we never saw before, sometimes because of vegetative cover. Have you discovered any really cool new things with looking at soil color maps? Has something popped out, an observance that you weren't aware of before?
Well, it's a good question. I mean, there's two things that pop out. One, I found I've been able to learn about places I've never been to before, like this one here in Alabama, by looking at the soil color maps. It's also a great way to find discrepancies, correlation problems waiting to be fixed, update projects waiting to be done, when you find just kind of blatant discontinuities at political boundaries. So I can't say that I've necessarily learned anything new because of LiDAR, but looking at these maps basically brings up, you know, many, many questions and to-do items for the near future.
Great. Well, I'm not seeing any other questions, so thank you for your presentation, and we'll move on to the next one.
Great, thank you. Let's see if I can
First,
get this.
first, I want to remind folks that the next webinar is going to be January 10th. So again, go through, look at some of your favorites, volunteer for an upcoming one beyond January 10th. Our next presentation is going to be by Tiffany Carter, who is a research scientist at the National Soil Survey Center in Lincoln, Nebraska. And Tiffany will be speaking on using combustion analysis to simultaneously measure soil organic and inorganic carbon. Tiffany, the floor is yours.
Thank you, Dave. Can you all see my slides?
Yes.
Great. Well, good afternoon again, everyone. My name is Tiffany Carter. I am a research soil scientist here at the National Soil Survey Center. The title of my presentation, as Dave has already mentioned, is using combustion analysis to simultaneously measure soil organic and inorganic carbon, specifically in one step. Before I get started, I want to take this opportunity to just quickly acknowledge my co-authors, Crystal Schecker, Steve Monteith, and Rich Ferguson. A lot of work went into this presentation, and so this is the result of a collaborative project between KSSL and the research staff. So I just wanted to make sure I acknowledged everyone that was working with us on that. Well, let's get started. Okay, so in general, soil science recognizes two primary forms of carbon, and that's organic carbon and inorganic carbon. The organic carbon is further divided into two groups. For the purpose of this presentation, we're going to refer to them as easily oxidized by combustion, which would be any of our organic carbon that's specifically derived from like plant residue, animal residue, root exudates, microbial biomass, etc. And then the other group will be our less easily oxidized by combustion. This is more of our elemental carbon, primarily like coal-related materials or black carbon, if you will. And then the other major group of carbon that soil science recognizes is, of course, our inorganic carbon. And so this is primarily calcium carbonate, but it could include other forms of carbonate, such as dolomite. And so again, for the purpose of this presentation, we're going to simply call it soil inorganic carbon. Now, as you're probably aware, there's been some increased interest in the applications of soil health and carbon sequestration. So there's quite a bit of interest in forms of carbon right now. And so the measurement and evaluation of organic and inorganic carbon levels really is of longstanding interest to quite a few. And so what we did here was take a look at how we can measure some of that carbon. I'll start by giving a little bit of background. Currently, KSSL has established methods for how they do carbon analysis. The total carbon, which includes both the organic and inorganic carbon, is measured currently by dry combustion. And so I'll spare you all the details, but there's a couple of important things to note. Here, when we're talking about total carbon, again, there's a combustion method that the temperature is heated up to over 1100 degrees. And so all of the carbon that's present there undergoes some sort of decomposition and is measured by a thermal conductivity detector. The inorganic carbon that's part of this total carbon is actually estimated from the measured calcium carbonate equivalent. And so the way that works is the calcium carbonate equivalent is measured by manocalcimetry. The inorganic carbon is actually estimated as 12% of that calcium carbonate equivalent. And that's because not all carbonates are actually calcium carbonate. The amount of organic carbon, to kind of break down from the total carbon to get those pools of inorganic and organic, that organic carbon, we've already talked about inorganic, is actually estimated as the difference between that measured total carbon that we had at the beginning from the combustion analysis, and then that estimated inorganic carbon that was from that measured calcium carbonate equivalent. And so I said all of that to say there's a little bit of a shortcoming with this method, and that is that both our inorganic and organic carbon pools are estimated rather than directly measured. And so what we did was test some alternative methods to see if we could do a little bit of a better job of getting some of those pools separated out. What we did was test alternative dry combustion methods for the simultaneous measurement of organic and inorganic carbon pools. The ultimate goal, again, was to measure and not estimate our inorganic and organic carbon forms in a more efficient manner. And so the new dry combustion method that we were looking at actually comprises two variants. The first variant, we'll call this variant 1, or our non-gas switching variant, uses oxygen throughout the analysis. It starts out at 150 degrees Celsius. We've got our sample in there. It heats up, ramps at about 70 degrees per minute, gets up to 400 degrees, and then holds there at 2 minutes. At this point, any of our easily oxidized organic carbon, again, under oxygen, should oxidize. And so it is picked up by the detector. And then after it's been holding for 2 minutes, things continue. It starts to ramp up again from 400 to 600 degrees, at which point our less easily oxidized soil organic carbon goes ahead to complete its oxidation. And then After it gets to about 600 degrees, our ramping continues up to 900 degrees, where our soil inorganic carbon goes ahead and thermalizes at that point. And so down here at the bottom of the screen, you can see we have, I'm not sure if you can see my mouse, but here, these little peaks that are here indicate our different pools of carbon. And so this would be our easily oxidized organic carbon, then our less easily oxidized organic carbon. And then here, finally, the peak would be our soil inorganic carbon. And then the sum of each of these three parts would be our total carbon that's present in this particular sample. And so again, all of this happens in the presence of oxygen, and that's an important note. Now, the second variant we're going to call our gas switching method. It has a similar process as the first. Starts out at 150 degrees, ramps up to 400 in the presence of oxygen. However, once we get to this 2-minute holding point, the oxygen, the machine that we're using here, shuts the oxygen off and uses nitrogen as a carrier gas. And so what this does is stops the oxidation of the organic carbon that's going on and allows for all of our soil inorganic carbon to go ahead and thermalize. And it ramps it up from that 400 all the way to 900 degrees and holds. Now, once we hit that 900 degree point, what the machine does is the oxygen is reintroduced into the atmosphere. And then at that point, all of our less easily oxidized soil organic carbon completes its oxidation process. And so it's a little bit different than the previous variant in that our different pools of carbon are coming out at different points. So again, first, you'll see, if you look down here at our little teeny tiny spectrum, first you have the easily oxidized organic carbon, then our soil inorganic carbon coming out, and then finally, any of that less easily oxidized organic carbon there at the end. And so in order to evaluate these variants, what we did was there were 110 soil samples that were selected. The samples that were selected include a wide range of soil textures, different carbon contents, including both organic and inorganic carbon, several other chemical properties, different soil mineralogies, and several taxonomic classes. They're from several different locations. So we've got a little map here, and all of the states that are highlighted in blue represent locations, represent states that some of our samples were taken from. Within these samples, it's also important to note that 32 of them were considered to be calcareous soils, and then 78 of them were considered to be non-calcareous soils. And so the idea here was to compare the legacy KSSL method, or the method that KSSL is currently using, with some of these new variants that I just discussed. And so all of the legacy data was taken from KSSL analysis that had been done previously using those current methods for both the total carbon and then the calcium carbonate equivalent. All of our samples were analyzed in duplicate using both the gas switching and non-gas switching method variants, and then ultimately compared with legacy carbon data. And so when we did that, we had some immediate observations. First, for all of our soils, both of our method variants were able to give us fairly accurate total carbon data with respect to the legacy combustion analysis. And so this was a good thing. When we were talking about those 32 calcareous soils, when we used our non-gas switching method, we found that it tended to overestimate that less easily oxidized organic carbon and underestimate our soil inorganic carbon. The reason for this is because there was some CO2 peak overlap. And so the machine using the non-gas switching had a hard time kind of differentiating between those two pools. And so they would kind of bleed together. And when we used the gas switching method for these calcareous samples, we were able to get accurate soil inorganic carbon readings without overestimating that less easily oxidized soil organic carbon. Now with those other samples, the excuse me, the 78 that were non-calcareous, when we used our gas switching method, we yielded some soil inorganic carbon readings that weren't as accurate. However, with those non-calcareous samples, when we used the non-gas switching method, we were able to yield soil inorganic carbon readings that had a teeny tiny amount of that soil inorganic carbon that was variable from what we were used to with the legacy method. And so, based on our initial observations, we had some provisional conclusions. Those were, first, to get the most accurate soil inorganic carbon and that less easily oxidized organic carbon. Those seem to be kind of the problem pools, if you will. And so to get the most accurate information for those, we should use the gas switching method for any samples that we determine to be calcareous, and then use the non-gas switching method for any that we determine to be non-calcareous. And the way to kind of decide which method to use with your sample would be to do a simple effervescence test and use that as the decision tool. And so with that said, our next steps from that were to kind of combine our data and look at things a little bit further and perform some additional data analysis, which we did. And so looking at how we've analyzed some of that data, again, this is kind of a repeat of what we saw from our provisional information. For total carbon, when we compared our legacy method to these new methods, using all of our samples, we were able to essentially get accurate total carbon. And so that is seen here. We've got good numbers here. Now with the organic carbon, same thing. We didn't see much of a difference between the legacy method and the new measured methods. However, as we discussed earlier, there was some overlap with our calcareous, or excuse me, when we were using our 32 samples that were calcareous. And so when we looked at the legacy method and then some of our methods that were derived, or excuse me, looked at the legacy method compared to these particular samples, we saw that we had some inaccuracies there. And so when looking at this, for the purpose of comparing apples with apples, what we did with our legacy data, and then again, comparing to our new method data, we converted the new method inorganic carbon data to our idealized, or strictly speaking, estimated calcium carbonate equivalent. We could have done this on an inorganic carbon basis, but the results would have been about the same. And so again, just as a reminder, I'm talking about our 32 samples that had measurable calcium carbonate, or those that we consider to be calcareous. Now, again, while all of our methods yielded data that were comparable, the question that came to our mind is, okay, which of these is absolutely most accurate so that we can make sure that we are differentiating between these pools correctly? And so what we decided to do to just confirm and make sure that we were on the right track was to compare them again to see which had the best relationship with the MIR spectra. This is because they're known to have, the MIR spectra is known to have spectral features that are associated with carbonate species. And so as we're doing this, there are a couple things that should be noted. First, the MIR spectrometer was calibrated using each set of reference data. And then second, all of the calibrations were built and tested using the cross-validation approach to make sure that we were being as accurate as possible. Okay, before I get into what things looked like when we compared to the MIR spectra, I want to paint a little bit of a picture to help understand what's going on with that MIR validation. And this is just in case there's anybody out there like me that isn't totally familiar with MIR. So what I want to use is pH, something we're all familiar with, just kind of as an analogy. So again, we're all familiar with the pH meter. The way it works, we all know, but essentially we grab some calibration solutions. Usually they're labeled maybe like 4, 7, and 9 or 10 as a pH. And so we calibrate our pH meter and we draw a standard curve. And if everything is accurate, we should end up with a straight line, kind of like this blue one here. We have a reading for 4, for 7, and then approximately 9. Everything lines up correctly. But it's important to note that the pH meter does not know what the pH of these calibration solutions are labeled as. And so what if those calibration solutions are mislabeled? If the calibration solution pH is off from what's supposed to be labeled on the bottles, then of course the millivolt readings on the instrument are going to vary accordingly, and we won't get that perfect straight line we're looking for. And so this red line here is an example of, say those readings are a little bit off, we're still able to come up with essentially this straight line, but the readings here don't align perfectly with our line that we've drawn here. And so just as a pH meter can be calibrated to measure the pH of our unknown solutions, it's known and established that an infrared spectrometer can be calibrated to estimate calcium carbonate. The reason is because the spectral features associated with carbonate are really well pronounced in an infrared spectrum. And so a statistically and useful calibration relationship can actually be established between the rise and fall of associated spectral features and the rise and fall of conventionally measured calcium carbonate equivalent. This slide here just has one example of an MIR spectra. Right here in the middle is an example of one of those spectral features that is associated commonly with carbonate. There are several others that are uniquely associated with carbonate throughout the spectrum. And again, these features tend to change or rise and fall with the amount of carbonate. And so with a range of calcareous samples from low to high calcium carbonate equivalent, a partial least squares regression analysis can be used to kind of tease out a useful calibration relationship between the instrument responses of the infrared spectra and the conventionally measured calcium carbonate equivalent data. And so that's what we used to do our comparison. And so our performance statistics here, or kind of what we're seeing here, is essentially similar to what I showed with the line on the pH meter. So that was our analogy for how we were able to just kind of make sure that our data that we were getting was aligning correctly with this, or we were using the MIR calibration performance to just kind of test which of these methods was going to provide us with the most accurate information related to how we could get these different pools of carbon. And so our legacy data, as you can see, we've got all of our data points here as these little yellow diamonds. With our gas switching method, these are the green squares, and then our non-gas switching method are the little red triangles. And essentially, the one that is most accurate is the one that best aligns all of its data with this line. And so as you can see, our gas switching method for those calcareous samples that we're talking about, our gas switching method best aligns here. There's less variation. You can also see that by looking over at the R squared. The gas switching method provides a little bit closer to that ideal 1.0 or perfectly aligned with the line here. And so with that in mind, we are able to essentially support our provisional conclusion to get the most accurate soil inorganic carbon and that less easily oxidized soil organic carbon. To make sure that we're getting the accurate results, we need to first use our gas switching method for any samples that we determine to be calcareous. Second, we should use the non-gas switching method for any samples that are not calcareous. And the way that we make the decision between which of these methods we should use is to just use a simple effervescence test as our decision tool. What we plan to do next with this is continue discussing it, working with it, and get started on kind of writing it up as a paper. Hopefully this will be something that can be useful to KSSL in the future, and then of course to the research lab. It does save a little bit of time, and it does not require it's not as labor intensive as the current method that's being used. And now that we have done this kind of small project and we know that we can get accurate data, this would save quite a bit of time for our technicians who are already working really, really hard with what they do there at KSSL. So this might be something that we could propose onboarding in the future. Again, I want to acknowledge those that have worked with me on this, Crystal, Rich, and Steve from KSSL. Everyone in KSSL for being patient with me as I'm running in and out of their lab and asking questions. I also want to thank everyone else from our research branch, my supervisor, Sky Wills, the lab director, Deb Harms, and everyone else who has even listened. You know, this was a lot of work. It took several months to get to this point, and so I just really appreciate everyone's support as we worked on this. The instrument here that I just kind of called it the instrument throughout the presentation, but it is a solid top cube. But I want to be clear that we are citing which instrument we used just for informational purposes, but this does not imply any sort of endorsement, and one should not be inferred. With that, I thank you for giving me a little bit of your time today, and I would be happy to take a couple questions.
Thank you, Tiffany. When, in interest of saving laboratory time, as you mentioned, would you recommend a laboratory test for effervescence, or is a field description of the effervescence presence or not going to be good enough?
That's a good question, Dave. Quite honestly, I think the field test should be enough, because what we need to do essentially, and I think that just kind of a visual, is there or is there not carbonate present, should give us what we need so that we know which method to send it under.
Okay. Another question, is there currently a way to differentiate EO SOC and LEO SOC in SPSD products?
That's a good question. The way, I guess the short answer to that would be not exactly. The way that KSS, and please someone from KSSL correct me if I'm wrong, but the way that essentially this is measured is you have your organic carbon and your inorganic. I'm not sure that KSSL at this time is trying to further separate those tools. It's not as simple as, it's not quite that simple. And so using this machine does help with that.
Okay, thank you. Yeah, keep that last slide up. Nice to see the pictures and the thank you. Not seeing any other questions coming in. Oh, yes, there's one. What is the size of the instrument, the soil talk that you had on the, I believe, the previous slide?
You mean like physical size, like how much space does it take up?
Yeah, that's like a meter by a meter, or are those like centimeter by a centimeter? What? How big are we talking for that thing?
Oh, it could certainly fit in in about a meter by meter space. The other thing to keep in mind is that there are gases that are attached to it. So it certainly fits on the lab bench. I'm sorry, I could have put an image of it in our lab. So it does not have a large footprint. It's a fairly small cube, but the thing to note would be, making sure that it's connected to its gases, which will take up some space. And then also it is controlled by a computer, which will also take up some space. But the cube itself will fit into a meter by meter space. Sure.
Okay, thank you. And just a reminder to folks that as questions come in that we might not have time for our presenters to answer, they will be answered and posted on the YouTube channel. So that's the end of the questions. So thank you, Tiffany, for your presentation. Thank you again, Dylan, for your presentation. I thank all the people that attended. We had over 130, I think, folks attending today. Glad to have you all here for number 20, and we look forward to number 21 next month. Have a good rest of your week. Bye now.